Good. Hello, my name is Lauren Connolly, and I am presenting an experiment done on the resistance of Listeria monocytogenes biofilms on sanitizing agents in a simulated food processing environment. Listeria monocytogenes is a pathogenic bacteria causing fatal infection known as listeriosis. Listeria monocytogenes is a facultative anaerobe, therefore it thrives under cold temperatures with or without oxygen. It is a small coccal gram-positive bacteria occurring in small chains. Listeria monocytogenes can infect the brain, spinal cord membranes, and bloodstream of the host via ingestion of contaminated foods, like unpasteurized dairy products like cheese or milk. Listeria monocytogenes infects a living organism by inducing macrophage phagocytotic uptake, where it displays D-glucose adhesion cells and other products to form adhesions from the cell to macrophage receptors. Through adhesions, Listeria monocytogenes invades the cell via a zipper mechanism to be fully phagocytosed. Now, once it is fully phagocytosed, the Listeria is encapsulated in the host. Listeria monocytogenes then lyses the vacuole's entire membrane with an exotoxin. Listeria then replicates in the cell's cytoplasm. It navigates to infect surrounding viable host cells by flagellar-driven motility known as tumbling. Listeria monocytogenes is one of the most fatal foodborne pathogens and is at the forefront of bacterial research today. In this study done by Dr. Pan, Dr. Brait, and Dr. Catherou, Listeria monocytogenes biofilms resistance to sanitizing agents was evaluated under a simulating food processing environment in order to better understand how and why Listeria monocytogenes can form such strong resistance to various sanitizing products. In order to prepare the Listeria monocytogenes for the experiment, the strains, 5 of them exactly, were stored at 80 degrees Celsius in a bactocryptic soy broth with 16% glycerol. Cultures were then inoculated from frozen stock onto plates at 27 degrees Celsius for 48 hours. Overnight cultures of each strain were inoculated at 37 degrees Celsius for 18 to 20 hours. Subcultures of this were transferred and also inoculated at 37 degrees Celsius for 18 to 20 hours. Bacteria was then washed twice and centrifuged for 10 minutes at 10 degrees Celsius while being suspended in saline solution. Once the 5-strain mix was prepared, then the biofilm surface was also prepared. The 2 different surfaces used for the biofilm are coupons, and there are the Teflon coupon and the stainless steel coupon. Both Teflon and stainless steel coupons were put in 25% nitric acid solution for 8 hours at room temperature. The coupons were then cleaned in detergent solution for 1 hour to rid of debris and excess things that could dilute the experiment and cause misread or inaccurate data. Coupon surfaces were rinsed in DI water and sterilized with steam for 15 minutes. After this, the biofilms were then formed on each kind of the coupon, both the Teflon and the stainless steel. They were repeatedly subjected to a 24-hour cycle, simulating the stress that Listeria monocytogenes would undergo in a food processing plant. So first, the coupons were exposed to a sanitizing agent for about 60 seconds, and this also contributed the control. This was the control. Then the coupons were stored in a sterile plastic tube with no nutrients or water for 15 hours. Then the coupons were incubated in a diluted growth medium for 8 hours. So if you put this all together, it's roughly a 24-hour cycle, and this was induced every 24 hours for roughly 3 weeks. Readings were taken before the process, so before they were treated to create a control. And then after they were treated, day 3, day 5, day 10, day 14, and day 21 of the 3-week, 24-hour cycle that is simulated, that simulates a food processing plant. So ultimately, the results display that the bacteria on The coupons reduced in number in the first week of the simulated food processing cycle, but they finally adapted to the stressful conditions and formed a resistance to the sanitizing agents. Biofilms that were repeatedly exposed to the peroxide sanitizer in the simulated food processing regimen developed a resistance to the peroxide sanitizer, along with other sanitizers like quaternary compounds and chlorine. Surprisingly, some cells that were removed from the biofilms on the peroxide-treated control coupons were not significantly different in their resistance to sanitizing agents. There's very few variation between the control and the actual treated, which is bizarre. The trends, you can look at figure 2 on my actual slide thing, and it displays the viable cell counts from the stainless steel and on the Teflon coupons during the 3-week simulated food processing system. There's a yellow line that represents the control and a blue line that represents the treated. So when you look at the linear regression lines, there's trends in both of them. So there's the Teflon coupon and the stainless steel coupon, and then control and treated. But when you look at the linear regression of each, the deviation between the control and the treated coupons for both coupons, didn't matter which one, their linear regression line, each of them had a y-intercept between the control and treated that was very small, so that can conclude that both of the treated and untreated with sanitizing agent coupons, they behaved in a similar manner when subjected to equal amounts of stress in the simulating food processing 24-hour cycle. So the analyzed data suggests that resistance of the treated biofilms to sanitizing agents may be due to attributes of extracellular polymeric substances. The extracellular polymeric substance is basically a compound that is secreted by the Listeria monocytogenes, or any microorganism at that, into its surrounding environment. Polymeric substances are established, and they basically establish the whole structural and functional integrity of a biofilm and are considered the fundamental component of the physiochemical attributes of a biofilm. So basically, the integrity of the biofilm is completely attributed to the extracellular polymeric substances. My bad. So, in conclusion, I mean, ultimately, the research on the bacteria Listeria monocytogenes behavior under specific conditions must be further induced in order to fully understand how actually Listeria is capable of forming this resistance with its biofilm to its surroundings, like what is going on with its extracellular polymeric substances, basically, and how is it strong enough to fight off what a sanitizing agent is attempting to induce. So the compounds secreted by Listeria monocytogenes are clearly receptive and capable of noting destruction, and furthermore can create a strong resistance to imposed sanitizing agents. But Listeria monocytogenes is an extremely fatal and pathogenic bacterium that infects by invading, lysing, and ultimately growing in a bloodstream, spinal column, or brain. So to prevent that, further research and the gain in knowledge of Listeria monocytogenes behavior is required to more greatly understand and hopefully prevent the presence of Listeria monocytogenes in a food processing plant, so people don't get sick and listeriosis can be avoided. So that is my project, and thank you, Micro, for teaching me about what happens at a cellular level. Thank you. Perfect.